1.
Buenos días, buenas tardes y buenas noches.
Hello, good morning, good evening, everyone, and welcome everyone who's joining us in this first Latin American Summit on Longevity. My name is Cristobal Thompson. I'm the Executive Director of AMIIF, the Mexican Association of Pharmaceutical Research Industries. And together with Margaretta Colangelo, CEO of Jthereum in California, and Héctor Valle and Santiago Valle, President of Fonsalud, we are creating this first meeting in Latin America. So let me tell you that worldwide, today we have over 1,000 people over 65 years of age. And as decades go by, there's no question that life expectancy will still grow. There are a lot of people that would get to 100 years or more. And so far around the world, there have been some very advanced treatments, and there's still some progress done in order to improve the quality of life of people. But now there's the time to make progress in another pathway, which is longevity, where today our experts will be sharing with us all the advances, how with this new vision of longevity, we can delay aging, which will allow people to live for longer with a better quality of life. So without further ado, I would like to switch to English so that I can welcome Margaretta Colangelo. Hello, Margaretta.
From more than 34 countries that are joining us for this first Latin American Forum on Longevity. It's a great pleasure to present Margaretta Colangelo, co-founder and CEO of Jthereum in California. I had the great opportunity of having a one-on-one with Margaretta during our Week of Innovation in March. And after that meeting, we agreed with Margaretta we should organize the first conference. And we said November 11th, and here we are. And for me, it's a great pleasure to have here Margaretta, and welcome to this conference.
Thank you, Chris. Welcome everyone to this great conference. I'm so happy that we're doing this and that so many people have registered to watch this. There's people from all over Latin America and from 34 countries, as Chris said. And, you know, I follow longevity very closely, and I've really started to hone in on the companies that I think are the most have the most potential to actually deliver treatments. And there's about 20 that I cover very, very closely. And these 4 scientists are in the very, very top of the companies in the whole world. And, you know, every time I talk to someone about the future, whether they work on Wall Street or they work in a high-tech company, I tell them about what these scientists are working on. And they say to me, you've changed the way I think about the future. So that's what I hope all of you who are attending the conference today will take away from this, that the future is not going to be like the present. The future, there's so much potential in the future. And many of you have not ever been exposed to the ideas that these scientists are going to tell you about today. But I am so fortunate. I get to read about them every single day, and I know them, and I'm really happy to share their work with you. So today we have the 4 scientists. We're going to start with Matt Schultz. He's from Oisín Biotechnologies. And then Greg Fahy. He's from Intervene Immune, and he runs the TRIM trials, which are really exciting. And then Johnny Bear from the Albert Einstein College of Medicine in New York, and Doug Ethell from Lucadia Therapeutics. So let's start now with Matt. Matt, welcome.
Thank you. I'm going to put up my slides here so I don't get stuck doing it while I'm talking. All right. Everyone see this? All right. So, yeah, my name is Matthew Schultz. I'm the CEO of Oisín Biotechnologies. We're an aging company based out of Seattle, Washington. And I got into this space in general from kind of an unusual background. I'd studied computer science and physics, actually. And the North Star, in fact, that brought me into the world of biology was that the essence of life is information. And chemistry is really just the substrate. In some respects, life is this ultimate bridge between, atoms and bits, information and chemistry. But particularly as it relates to aging, there's real limitations to small molecule drugs. I mean, you think about the history of medicine has been largely plants and prayers since the dawn of recorded history. And although we have gotten mileage out of that, there's, real issues with it. I mean, that's why most drugs you take have a list of side effects that's, a mile long. And in aging, this problem becomes particularly acute in that, I mean, you can never shovel enough pills down your throat to undo aging. Too much changes. But if you begin to be able to manipulate the code of life, you can do all sorts of things that really wouldn't have been possible before. And so the reason we're going after aging kind of specifically is that, it's a master corollary for pretty much any malady you could think of. I mean, you have worse odds of having cancer as a clean living 70-year-old than you do a chain smoking 20-year-old. And that's not to say that your ill-spent youth isn't going to haunt you later, but it's just that aging itself is the dominant risk factor for almost everything. And so if you want to be able to tackle, all these things in human health, going after aging itself has a lot of value. And the first tool we had built actually was a senolytic. And I won't beat the horse about what senolytics are too much. I mean, they're damaged cells and want to kill them. But what really had launched this effort was a couple of mice that made it to the New York Times. This is a picture from a study done by the Buck in the Mayo Clinic, and lots of people have seen this, I think. But the takeaway is those mice are litter mates. They had the same diet, the same lifestyle, the exact same genetics. The only difference was that they removed those damaged cells in the healthy-looking mouse and did not in the sad-looking mouse. And so you can trace, most of the pathologies of age to senescence in some form or another. And obviously, it's not the only factor, in that if you remove them, it doesn't make, immortal mice. But they do live about 25% longer, half as likely to get cancer. And if you treat them from youth, most of these pathologies of age don't manifest themselves at all. And so this is really exciting. But I guess in the tradition of these academic studies, it was useless translationally. I mean, you'd have to go back in time and modify your own embryo to do this. And it's obviously not a viable treatment. So Oisín really got its start trying to do this in a way that was clinically viable. And to do that, we really needed a new class of genetic medicine. And so people have wanted to mess with DNA in the body ever since they knew what double helix was. But the body has a vested interest in not letting you do that. I mean, it thinks virus right out of the gate, and there's lots of defenses against it. And indeed, though, viral vectors have been kind of a dominant tool for manipulating information in life for a long time. I mean, they're purpose-built to do that. But they have major issues in terms of they're quite expensive to manufacture. They're obviously very immunogenic. Your body will mount a response against them very quickly, and often prevents redosing. And for a scalability point of view, having treatments that cost $1 million a pop is not great, especially when addressing a market like aging. And lipid nanoparticles are kind of the other tool for doing this. And they had kind of fallen out of favor years ago, higher profile failures with companies like Moderna, and now are back into the spotlight as rock stars because of successes with companies like Moderna. And these are basically little fat bubbles that carry DNA or RNA. And they're relatively cheap to make at scale, but they had major toxicity issues. In fact, one of the reasons these companies weren't faring super well a few years back was that if you administer enough of it to make changes throughout the body, it tends to destroy the liver. And that's just because of how they accumulate. They make a Faustian bargain of sorts with their chemistry, in that if they're neutral, they work fine, but if they have a charge, they become toxic. Unfortunately, if they're neutral, they also don't work. So it has limited their dosing. And so we built this tool called a proteolipid vehicle, which is basically the same kind of fat bubble, but neutrally charged. And it gets into the cells this little fast protein. And it's super cool, and I don't have time to talk about it in detail today, but it first came from a virus that infects the stomachs of alligators and birds. And what's special about it is it's tiny. It's about 100 times smaller than a fusion protein from, say, like COVID or the flu. And it can do something really tricky in that it will form a complex and flip, and it mixes the lipids here of this particle with the lipid of a cell. And so it can dump a payload into any cell in the body. It has no tropism, and it's not immunogenic. And I mention all that because the next few slides kind of depend on this core thesis, like information in the body and delivery that involves targeting with information instead of chemistry. And so this next slide here, these are little videos. These are senescent cells, and we make them glow green so you can see them. And what we did to get rid of these, instead of using like a poison or something like a chemotherapy to get rid of them, we said, hey, how about we write a little program in DNA that says, if you're senescent, kill yourself. Because it might sound odd, but a damaged cell knows it's damaged. And so we administer that and you activate it. You can watch the cells self-destruct in front of your very eyes. It's pretty cool. Time-lapse microscopy. They're going into apoptosis. You can run them out until they all die. But what's more important is actually this one on the right. Here you have mixed cells, healthy cells in red and the green cells being senescent. And when you activate it, you can kill just the damaged cells and not hurt the cells around it. And this is really important because traditionally when you think of killing cells, you think chemotherapy. But the standard for cancer might be eating poison, and people might tolerate that because they're going to die if they don't. But there's no way you're going to feed a bunch of otherwise healthy old people poison. And there's also no way that you can kill like one bad cell in an ocean of good cells and not cause collateral damage if you use a poison. The only way to be that selective is to not use the drug at all. So in this case, there's no drug. It's DNA, computer program in a fat bubble. And it allows you to do really cool things in like a body-wide context. So in this case, we're making firefly protein called luciferase, makes fireflies' butts light up. And we injected IV into this mouse, DNA that encodes it. You can see it goes all through the mouse lights up. If you look at its organs, they all light up. But what's really interesting is here on the right, this mouse got the exact same dose and the exact same route of administration, tail vein. But now we gave it that firefly protein under a program that says only turn on in a cancerous cell. And there's a tumor implanted in this mouse in this little red line here. And you can see that all of that expression is restricted to the tumor. Like all these tissues got the exact same dose, it just didn't turn on. So what we've done here really is we've taken targeting out of the realm of chemistry and we brought it into the realm of information. And you think your body has exquisite control to kind of control every aspect of cellular life. We can actually manipulate it or ask it a question. It also allows you to do some kind of more interesting things beyond simply killing cells, like building muscle, work on building, a nap store for the human body. I don't know if you've seen this cow before. It's a Belgian blue cow, a naturally occurring mutant that's about twice the muscle mass. It's that way because it doesn't have a brake on muscle development. It's missing a gene called myostatin. And people have done this with mice, but you can take it a little further. You can say, instead of just knocking out the brake, what if we hit the gas? We basically tell the body it's exercising all the time when it's not. Make this molecule follistatin. It actually gets times the size in that case, this monstrous mouse. But this is really, the biochemical pathway for skeletal muscle exercise. But it's also, a computer program written in DNA. We just tell the mouse, get stronger. And these all were modified from birth again. And if you do it when they're older, it's not quite so extreme. So this is one that we actually built. And these mice get treated when they're adults. And it's just a simple injection. You get one, so infusion IV. And you go forward less than a year later, they have 50% greater mass and they're double the strength. And that's pretty wild, right? I mean, consider like I weigh about 200 pounds. That'd be adding 100 pounds of muscle. It's absolutely enormous. And other than making athletics a bit more interesting, I think that the real benefit for this is treating frailty with age, because physical strength is everything with old age. I mean, it's the difference between like a wheelchair and a cane, or a cane and jogging. Like falling over and breaking something. This is a huge aspect of it. And I just highlight this, a relatively simple use case, but we've gained the ability to manipulate information in the body, and it allows us to do some pretty wild things. And so as a company, we have a whole pipeline full of applications like this, with the holy grail being kind of in situ tissue regeneration indefinitely, like to make old things young again. And so that's what we're working on. And I think I'll leave it there. It's time.
Margaretta, you were muted.
Oh, here. Thanks, Matt. So, Matt, I know you have to, you have another talk after this, but do you,
Yes.
I could ask you a couple questions now, or we could wait for the other presentations, and at the very end we start with you. What do you prefer?
Either way is fine with me. I need to leave at 10 before the hour, so whatever's most convenient for you guys.
Let me just ask you a couple questions now, so because if we lose you, I won't be able to ask you.
All right.
Can you just talk a little bit about the mice that were treated, the male mice that were treated, and the bone density?
Yeah.
I love that.
So that was a study we did with the senolytics, so killing these damaged cells. And in that case, we treated mice starting in old age at 2 years of age. So kind of like, I think 70, 80 years old for a person. And once a month, we would just give them this thing to kill those damaged cells. And we saw something really interesting. The bone density actually went up over time. So a lab mouse has a survival rate of about 2.5 years. In fact, at 2.5 years, half should be dead, half should be alive. By 3 years, they should all be dead. And so when you start this at 2 and you're measuring them at approaching 3, and their bone density has actually gone up, it's pretty amazing. And it's not entirely clear what's doing that. We have theories about it, but basically, the core thesis about removing senescent cells is that your body has a substantial regenerative potential that's latent. It's being suppressed by the environment created by those senescent cells, their secretory factors, and that simply removing them allows your body to regenerate a great deal. But you can consider for a moment that the best drugs for osteoporosis, for the most part, they slow bone loss. To go up is actually really extreme. They were approaching the density of young mice by the time it ended.
The pictures, the photos of them are fantastic, where you can really see the bone density. And then one other thing is, could you just describe how quick it is? So, for example, you gave mice who had tumors the treatment, and then within 48 hours, there was...
Yeah, so the tool we're using, I had to limit my data due to time, but it's a suicide gene called inducible caspase 9, and this causes the cells to kill themselves. It's kind of like a cellular recycling program, I suppose, if you think about it. The cells need to kill themselves and be reused for lots of purposes. But when you turn it on, cell death begins to occur within minutes, basically. And so in this particular study, the tumor is about 90% reduction in size, excuse me, in only 48 hours. So it was
that's amazing. The tumor, and it was prostate cancer, correct?
Yeah, that's right.
Okay, so the tumor in a mouse was reduced by 90% in how many hours?
Well, most of the death occurred probably within the first 8, but we measured it at 48. So
okay, I love that. That's fantastic. Well, thank you so much. Stay on till the end, but I know you have to leave for your other conference. So if you do leave and before we finish the rest of the presentations, thank you so much for joining us. I think when people hear about what you're working on, it's just going to really change the way they think about the future, make them very, very excited about the future. Thanks, Matt. So the next scientist who we're going to hear from is Greg Fahy. And Greg, back in 2015, he and his team were the first people to show that it is actually possible to reverse aging in humans. And they did this during the original TRIM trials. And right now they're conducting their second group of TRIM trials. It's called TRIMX. So that's just my little introduction. But Greg, welcome. And thank you for being here. You're on mute.
Okay. All right. So let's see if we can get the screen shared at this point. Okay. All right. Thank you. Let's go back. Okay. So I'll be talking about Intervene Immune. It's the company that has been running the trials that we were just hearing about from Margaretta. Our goal is to find ways of intervening in human aging sooner rather than later, because even things that age very well can hope to do a bit better. So our inspiration is to go after this goal of thymus regeneration. The reason we're interested in this is that the thymus, as many of you will know, is the master gland of the immune system. Its job is to manufacture immunocompetent T cells, which combat various invaders, infectious disease and cancer that tend to kill us more and more frequently as we get older. The thymus is located in the chest cavity underneath the breastbone. It's depicted on the left as this glowing yellow ball, but on to the right of that, what you see is a series of images showing that the thymus begins with a lot of functional mass, which is competent for making T cells. That's the pink area. But over time, that mass is replaced more and more by fat until by about the age of 70, we have very little functional mass left. And what that leads to is shown on the lower left, which is that the sum total of all of the antigens that the T cell population is able to recognize is foreign, declines by about 98% between the ages of 60 and 80. And because of that, our susceptibility to infectious disease and cancer begins to skyrocket within that same time frame. And what you see on the extreme right bottom is that if you look at the totality of infectious disease mortality and cancer mortality as a percentage of total deaths, it rises from about 5% when we're around 20 to about a third of all deaths when you get to be about 60 or a little, even a little bit younger than that. So regenerating the thymus so that we can have healthy T cells is imperative. And if we can do this, it'll have major effects not only on longevity, but on well-being and morbidity. So we did a trial called the TRIM trial. It was based at Stanford University. Trial ran from 2015 to 2017. It involved using growth hormone to regrow the human thymus. But growth hormone has a side effect, which is that it raises insulin levels. And so in order to combat that side effect and thereby get a better effect on the thymus, perhaps, we used DHEA and metformin to lower the insulin. The experiment was only done on 9 people. They were healthy. They were aged 51 to 65. You see the first 6 men in the middle of the slide. That's the cohort 1. And the last 3 men on the right, cohort 2. We gave them the treatment 4 times a week for 1 year, and it was individualized to each person. We were able to publish the study in 2019 in Aging Cell. So what we observed is partly what we expected, partly what we did not expect. What we expected, or at least hoped for, was radiographic evidence of thymus regeneration. So the images show the thymus of one of our volunteers before and after 9 months of treatment. You can see that the thymus is getting less white and more dark. That signifies the replacement of fat within the thymus with putatively functional tissue. And then to verify whether that tissue actually is functional or not, we looked at the appearance of naive T cells, newly derived T cells in the bloodstream, and we saw that the numbers of naive CD4 and CD8 cells went up, and recent thymic emigrants went up as well. One of the things we were concerned about was inflammation. As we get older, we become more inflamed. Since that's an immune function, we were warned before we did the study that maybe goosing up the immune system would cause inflammation to increase, but actually, at least based on CRP, inflammation went down. That may be significant because inflammation is probably related to atherosclerosis and most of the diseases of aging. We also saw something I was hoping to see, which is a clearance of senescent CD8 cells. These are PD-1 positive cells. These cells block the body's ability to reject cancer. So a reduction in the population of this senescent cell group may actually improve cancer surveillance. So some of the unexpected things that we saw are here. So one of the things that falls in that category is hair darkening. We saw it particularly in the example that I'm showing here, but we saw it also in a couple of other people in the trial. We had evidence that kidney function improved and maybe even continued to improve after the end of the treatment between days 365 and 340. We also saw some evidence that possibly our treatment is improving prostate health. That's signified by a decline in prostate-specific antigen, which happened very rapidly in the trial and for the most part persisted until the end of the trial, which is sort of an anti-carcinogenic effect. And then we saw supporting evidence based on some measurements that were made on CD38 positive monocytes at Stanford's Human Immune Monitoring Center, showing changes in these markers that suggest at least the possibility that we actually reduce the susceptibility of our population to virtually all forms of cancer and many diseases of aging, including all-cause mortality. So we wanted to see if these hints that maybe there was some generalized aging reversal going on could be substantiated. So Steve Horvath at UCLA, who invented the first good measurements of aging, the DNA methylation clock, applied that clock to our volunteers' blood samples. And panel A shows his original pan tissue clock showing statistically significant aging reversal after 12 months of treatment. So he applied 3 other clocks and saw essentially the same results. And the average response for all clocks is shown on the right in color. So on average, we reduced aging by about 2.5 years. And that was pretty exciting. Since the trial was completed, Steve actually went back and reanalyzed our blood test data. So the clocks that I'm showing at the top of the slide are all based on measuring DNA methylation changes, which requires that the samples be frozen and run all together at the end of the trial. Whereas the blood-borne clock, which is I call the plasma phenoage clock, can be done on the fly and it can be done retrospectively just based on the blood results that we got. So as you can see, the overall results using the plasma phenoage clock were pretty similar to what we got with the other clocks, and intermediate between the phenoage clock and the Grimm age clock, which are the best epigenetic aging predictors of future morbidity and mortality. So to summarize the results in the cartoon on the bottom right, normally when a year goes by, we get to be a year older. And there's been this theoretical concept of longevity escape velocity, which would occur if after 1 year, we've been able to reverse 1 year of treatment. 1 year of aging, so that there was no net aging over time. And what we saw is that we were able to accomplish that and then some, because on top of that, we added another 1.5 years of aging reversal for a total of 2.5. So these were pretty interesting results. So we decided to investigate them in more detail in an extension of the TRIM trial, the TRIM-X trial. We enrolled our first cohort last November. You can see that that cohort includes the first woman ever treated according to this plan. The plan for the trial is on clinicaltrials.gov, and we're enrolling people between 40 and 80 years of age. And we moved the venue from Stanford to the Lundquist Institute in Southern California, mostly for convenience. So far, we've enrolled 26 people. That includes 6 women and 2 untreated control men. We're still enrolling at this time. So the course of the trial is still early, it's still young, but we've seen some signs in some cases that we're able to confirm some of the results that we saw in TRIM, which is encouraging to us. For example, we've seen a reduction in CRP. We've seen reduced PSAs and really a large number of the men. We've seen improved kidney function. We've seen even early signs that biological aging, as measured at least by the plasma phenoage clock, is able to go in reverse in some of our volunteers, including, in some cases, very rapidly. We're also seeing some new effects. There are signs that people who have elevated carbon dioxide levels in the blood show a reduction in carbon dioxide, which suggests the possibility of improved lung function. And we're seeing in some people a reduction in blood urea nitrogen, suggesting that we're being able to pick up an anabolic effect of the treatment that way. So far, the side effects seem to be reasonably mild and manageable as they were in TRIM. So I'm just going to give you a little anecdote here from somebody who was in the original TRIM trial and then has started again on the TRIM-X trial about 5.3 years after he started TRIM. So at the start of TRIM, these are all plasma phenoage data. So the red dots are plasma phenoage, either measured or imputed. The green is chronological age, and then the blue is phenoage upon treatment. plasma phenoage upon treatment. So as you can see at the beginning of the trial, this gentleman was 3.7 years older than his chronological age based on the plasma phenoage clock. At the end of treatment, he was 4.4 years younger than his chronological age. If you add that up, that's about an 8.1 year gain in biological age, at least based on this clock. A little bit shy of a year after he stopped treatment, his biological age continued to go in reverse, such that he was 7.7 years below his chronological age, or 11.4 years below where he would have been if his plasma phenoage had just continued that upper line. When we started him on TRIM-X, 5.3 years after TRIM, his plasma phenoage had regressed a little bit back closer to his chronological age. But when we started him on TRIM-X, he very rapidly went to a level of 4.2 years below his chronological age, or 7.9 years below where he would have been if he had never been treated in any way. And that latter result happens to be 2.3 years younger than he was biologically when he started TRIM about 5.5 years earlier. So if we take that seriously, and we're not saying you can take it entirely seriously, but to the extent that the data have meaning, it looks like this particular man was able to have his age be less 5.5 years after TRIM than it had been at the beginning of TRIM, which is encouraging. So the next steps are we need to convert this into a marketable product. And so we're going to be meeting with the FDA to get their opinions about what endpoints we have to meet in order to get approval of our treatment for marketing in the United States. And there are issues of scalability. Our treatment involves a fairly intense amount of data processing, and we're working on that so that we can expand this to a large number of people. And of course, we need additional safety and efficacy data. And we're going to be developing new diagnostics and adding new diagnostics and clocks to further substantiate the treatment's effects. We're also going to be looking at improving the treatment in a variety of ways, both with respect to efficacy and safety. And of course, ultimately, we'd like to do some mechanistic studies. So the conclusion is that it seems like there really may be feasible to develop aging interventions that can have a meaningful impact on people in the near future, not in the far distant future, so that we can pursue goals such as the visionary goal that we're presenting at the bottom, because some people are just too valuable to allow to age. So we can be contacted at my email address for anyone who wants to work with us to try to achieve these goals. So thanks for your attention.
Thanks, Greg.
Okay.
And during the discussion time at the end, we'll talk more about the TRIM trials. And but thank you so much. It's so interesting. So the next scientist is Jean Hébert, and he's at the Albert Einstein College of Medicine in New York. And he's going to talk about brain aging and replacing brain aging and rejuvenating the brain. And he wrote this great book. It's not available in Spanish or Portuguese, is it, Jean?
It's actually being there is a translation. It's about to be launched in both Portuguese and Spanish. Spanish first. I'm not sure when the Portuguese.
Okay, fantastic. I'm really glad because I tell a lot of people about this book, and it's great. And so, okay, go ahead. I'm excited to hear your presentation.
Yeah, thanks for bringing that up. Very generous. The book is already outdated, though. I wrote it 1.5 years ago, and if I had to rewrite it, it would be a lot of it would have changed or improved in the prospects of this tissue replacement approach.
But you know what? It's a great introduction to what you're doing, and I really I'm supposed to write an article about it, but I've been so busy planning these summits that I haven't been able to. But when you publish it in Spanish and Portuguese, tell me, and we'll send it to everyone who attended the conference, like an announcement and a link so they can read it. Mm-hmm. Okay.
Yeah. Well, thanks. And thank you for having me here, and thanks to Chris and also to Cynthia for helping make this happen. So yeah, I'm Jean Hébert. I'm a professor at the Albert Einstein College of Medicine. I've been studying developmental biology my whole career. So how we're made, from a single cell to become a complex organism. And it's because of the idea that if we understand that well enough, we can recreate it later in life to, instead of generate tissues for the first time, regenerate them using stem cells, for example. So I'll start sharing my slides now. Okay, and hopefully you guys can see it. Okay, is that looking good? Okay. And so, I'm going to take a hard line here about my views on aging and how we're going to beat it. And I think brain tissue replacement can be viewed as a cornerstone to beating aging. And I'll tell you why I think that, and then we can discuss it afterwards. So first of all, what is aging? I know a lot of us define it quite differently, but I think it was well characterized by biochemists decades ago when they started looking at young and old tissue at a molecular level and found that basically every macromolecule, every type of macromolecule in the body accumulates sort of this stochastic damage over time. It's non-enzymatic damage, so it's not caused strictly speaking by biological processes. It's more physical and chemical processes that accumulate this damage. And so if we take a cell, for example, you can see a blue cell here, and it's surrounded by a matrix that is made of protein, largely protein and carbohydrates. That all accumulates, all those molecules accumulate damage over time. And as an example here in red are some of a subclass of forms of damage that occur to proteins, right? And this is well characterized that this happens over time in our bodies. And equally complex forms of damage accumulate to our other classes of macromolecules, DNA, lipids, and carbohydrates. And so, those are known to cause, at least for extracellular matrix damage, so that's the damage that surrounds cells, not actually in the cells, or DNA damage alone, are known to cause the other hallmarks of aging, which are therefore downstream and effects of these forms of macromolecular damage. So we need to address those forms of damage. And, Matt brought this up nicely in his intro about the limitations of drugs because they always have side effects. So trying to come up with, combinations of drugs or molecular treatments that can reverse these hundreds of forms of damage that occur differently to thousands of macromolecules without, having worse side effects, I don't think is plausible. And some of that damage is also irreversible in the sense that it's a loss of information, such as changes to DNA sequence or glycan sugar modifications. So, small molecules and even complex proteins are not smart enough to distinguish, what's a good covalent modification from a bad covalent modification. And so it's really, and don't have the information content to reverse that kind of damage. So this sounds rather bleak, right? I mean, if this is aging, what can we possibly do about it? And that's why biochemists stopped working on aging, because they were like, what can we do about this? But in the meantime, developmental biology, regenerative medicine has made a lot of strides. So I'm very optimistic that we can actually beat aging. And the way we're going to do it is by reversing or replacing the tissues of the body. Because when you replace a tissue, I don't think anybody would argue, but you remove all that damage, and then you put in a young, newly developed tissue that won't have that age-related damage. And, one of the fun things about writing the book was just reading up on all the progress that's been made in terms of cell, tissue, and organ replacements in humans. And so basically, a lot of progress has been made, and the feasibility of this approach, at least the surgical aspect of it, has already been demonstrated. Every part of the human body has been replaced. Unfortunately, not by lab-grown or manufactured organs and tissues yet. Most often from deceased donors, which is not because organs are in short supply for transplants, for example, they need to go to people that are in dire need due to disease. But these organs, every week, there's progress being made. We hear about new kidneys being tried in preclinical models, for example, other organs. So progress, it's inevitable that we will get to the point where we can replace every part of the body, except the brain. The brain is problematic. Obviously, we can't replace the brain as a whole organ. That's who we are. It defines our self-identity. But we can replace it, conceptually at least, and in practice, I think. Hopefully I can convince you of that. We can replace it progressively over time. And so the reason I think we can is because of how the brain works. It's super plastic in the sense that functions can move from one substrate to another without us noticing, without an interruption in function. So this is very well established in the neuroscience field. We've known this for a long time across mammalian species, across functions in the brain. This is one of my favorite examples because it has to do with language, which is very dear to us. But even a function as complex as language can move from one part of the brain to another without the individuals losing the ability to speak. If the original language center is very slowly destroyed, which in this case was due to benign gliomas, which take years to grow, but during that time, language moved to another part of the brain. And even when you remove the tumor with surrounding tissue for good measure, these individuals can still speak. And then if you look with electrodes, a functional MRI, you can see that language has moved to another part of the brain. So this plasticity means that in principle, at least, we can replace every part of the brain progressively over time. In practice, our lab and many others have shown that if you put in young, immature brain cells, they don't care that they're in an adult environment. They will do what they do during normal brain development when we're fetuses and infants. So they will differentiate into nerve cells that project to correct targets and electrophysiologically integrate. So they integrate in existing circuits. So we've shown this, and a dozen other groups have shown this. So here, what you're seeing on the left here is a lesion. Top left is we make these canonical lesions in a mouse brain as a model, and then we can fill it with these precursor cells which differentiate. What we've added to this is the ability to also include vascular precursor cells that form blood vessels that fuse with host vessels and circulate blood within the tissue. So this allows us to then use the mouse as a platform, not only for building mouse brain tissue, but also human brain tissue. Again, just to figure out how we can best do that. So this is a great step forward, but there's still a long ways to go. And I certainly don't want to make this sound like it's an easy thing. I think accomplishing functional brain tissue will be akin to what it was in the early 60s to put a man on the moon. It's going to be very challenging. But even then, we knew it was physically possible to do that. We just hadn't worked out the details yet. It's sort of the same thing here. There's a lot of details that need to be worked out, but there's no biological reason why we can't do that. And so one of the details is assembling the right components, the right cell types in the brain. There are many of them. They need to be there in the right ratios. We need to have them develop the way they normally develop in the embryo and fetus. But, we have a plan in place to do that. We think we'll be able to. And then, of course, we have to demonstrate the functionality of that in animals that are larger than rodents, because a rodent brain is quite small. And even though it's constructed the same way as a human brain, to some extent, size matters. So we would need to test this in a bigger brain as well for higher cognitive function. So we have plans for doing that as well. And of course, we want to use this for aging, but that will not be our first indication. There are many steps along the way where we can use this tissue for other things like disease modeling, drug testing, treating more localized damage in the brain, like stroke or localized traumatic brain injuries, and then move to dementia and aging. But, you know, we hope to do that before too long. And this is just, you know, a cartoon illustrating how this would be done, how we would use this tissue to reverse brain aging. You start with this nice, you know, beige colored brain, young brain. As you get old, it atrophies, creating space to add tissue, but you also need to remove old damaged tissue. And we can do that just like the example I gave with the slow growing benign glioma, except we're not going to use gliomas. We have ways of silencing neurons progressively over time, allowing those functions to seamlessly move to another part of the brain. And then once they're, you know, physiologically silenced, we can remove them and also replace that space with new tissue. And we do this reiteratively a few times, and we are back to a young brain that doesn't have any age-related damage. So, you know, we're not there yet, but that's in a cartoon what it's going to look like. So I think in combination with replacements for the other parts of the body, which a lot of people are working on in regenerative medicine, replacing the brain will allow us to beat aging. And of course, this is, you know, we have a group of people working on this, which is great, but we'll, you know, need to expand this quite a bit moving forward. So, yeah.
Thank you so much, John.
Thank you.
So, Matt, I think it's time you're going to have to leave soon for your next conference, correct? You're on mute.
Yes, unfortunately, I do.
Luckily, I asked you questions at the beginning. Yeah,
I know.
Thank you so much. You know, anybody who wants more information about Matt's work, you can go to the Oisín website, and there's a link on the Summit website. But also, there's many YouTube videos, and I watched them all with Matt talking about this, and they're really, they're really interesting. So I really encourage all of you to, if you're inspired by what Matt, you know, presented today, please watch his just search his name on YouTube. There's great videos.
Yeah, feel free to reach out on, you know, LinkedIn or email or anything like that. Always happy to talk about it.
Thank you, Matt.
Thank you very much, guys. It's a wonderful group.
And now the next scientist is Doug Ethell, and he's the founder of Lucadia Therapeutics. Hi, Doug. You're on mute.
Hi, Margaretta. Can you hear me?
Yes. Mm-hmm.
Okay, great. Thanks for the introduction. I'm going to share my screen right now. And okay, there we should be good. Oh, we're back one. So I'm Doug Ethell. I'm a neurobiologist by training. I worked on Alzheimer's disease as a professor for many years. And some of our technology got so relevant that I translated it into Lucadia Therapeutics and then shifted full-time to Lucadia a couple of years after that. Alzheimer's disease is kind of the holy grail of healthcare because there are 40 million patients worldwide and another 250 million expected in the next 30 years. We just published a paper showing that Alzheimer's begins as a CSF disorder, and we've got a way to fix it. So CSF, or cerebrospinal fluid, clears spaces between neurons to rejuvenate the brain. Thanks, Gene, for your introduction about extracellular spaces. So it really does that job in the brain. It starts in ventricles, shown here in blue, And the ventricles make about half a liter a day of CSF. From there, it moves outward, percolating through the tissue, picking up insoluble metabolites and debris and exosomes, carrying it all to the subarachnoid spaces on the surface of the brain. So this movie shows how the pulsing of the heartbeat actually shifts neural tissue. And many people in neuroscience don't really appreciate that there's so much through and out of the brain. Okay. The thing is, this porosity of the cribriform plate decreases with age. So here's a study with 330 subjects that we did, age 20 to 90. And if you notice the red trend line here, it starts to decrease in the 60s and 70s as the porosity is going away. Let's look at a heat map. Here's people 55 and under and people 65 and over. You can see where the apertures are, right? Red and purple being the most intense apertures. As you get older, they're sort of going away. Looking at that another way here on the bottom right, people under 55, the number of apertures is the white bar, and the gray bar is the total surface area of all the apertures that we showed. All right, people 65 and over have fewer apertures and a smaller total cross-sectional area. And people with Alzheimer's disease on the very right, they have a similar number of apertures to those over 65, but much smaller cross-sectional area. So it's really being choked off. Okay, this is great correlative data, but to show the mechanism, what we did was we blocked the cribriform plate in ferrets, and then we looked for behavior changes a few months later. Here on the bottom left, it's between 2 and 6 months of age. This is how ferrets did in a maze. It's a test of spatial temporal memory, which is one of the earliest things to go in Alzheimer's disease. That's why people get lost and the family spends hours trying to find them. So if we look at the dashed line, these are the control animals. And over that time, the amount of time it took them to solve the maze decreased because they're figuring it out. The solid line are the occluded ferrets. They took longer and longer. They had a progressive spatial memory deficit. Then we looked at the anatomy. Here's the underside of the brain. That red area that I showed you in the human brain, it's marked here in green on both of these. And the control side, it's about 40% bigger because it's smaller and smoother due to massive atrophy of this structure in the ferrets. So what we have is cribriform plate blockage as we get older. It's very extreme in Alzheimer's patients. If we experimentally block the cribriform plate in ferrets, we get Alzheimer's-like effect, early Alzheimer's effects. So it seems that what we've got here is a plumbing problem. And what we've done is developed a device to bypass the blockage, to restore the flow of CSF and help the brain rejuvenate itself. So the device is called Arethusa, and it goes in through the nose because the cribriform plate sits at the top of the nasal cavity. It's very accessible. Neurosurgeons will use the transnasal route to work on pituitary tumors, which sit back here. Okay, now we want to do a clinical study with this, but first we had to lay some groundwork. The first thing is to figure out who has the best possibility of benefiting from the device. So we need to know who's got MCI and which subjects we would want in our study. So to do that, we made a Fitbit for the brain, which we call it, and it's available in 22 languages on the Google App Store and on Apple's App Store. It's free to download and free to play. Have a look. Okay, so the icon on the top left is the Procogni memory tracker. It's available in Spanish, Espanol, and Portuguese. And one of the things that people can do is sort of play some games and puzzles and track how their memory's doing. And they can also take the Lucadia memory test, which is a great way to really test yourself. These other apps here are some of the fun games that we did for the Lucadia memory test. They're things like Sudoku, Infinite Mahjong, and Sudoku with icons. And again, those are all free and available on the stores. So physicians can use a HIPAA-compliant version of this to test 7 aspects of memory in patients. So our deep learning can use that to then figure out what their brain age is. So here, if we look on the y-axis, that's their brain age. And on the x-axis, that's their actual chronological age. Now, if you were perfect, you would be on this red line up here, right, where the two match. People who have older brains than they should have cluster up here. And these are people with Alzheimer's disease and dementia. And people down here have mild cognitive dementia. Now, 20% of seniors have mild cognitive dementia, but they don't all progress to Alzheimer's disease. This is a great way for doctors to get LMT results immediately and kind of figure out what they should do next. If it looks like they're up in that group, the next step would be to get a cribriform plate scan. Now, this can be done at a dentist's office in their own hometown. The scan itself takes about 20 seconds to do. And then Lucadia will use our deep learning algorithms to convert that into a good image so we can see the apertures and calculate the porosity of the cribriform plate for the subject. And then another type of deep learning we developed, machine learning, puts together that anatomical data with the memory data and some personal questions like, is there a family history of Alzheimer's disease? Have they ever broken their nose, which can damage the cribriform plate? And perhaps also some biometric data from smartwatches and smartphones. We put that together and provide the physician with an opportunity to see how they stack up in all of these aspects of memory, what our anatomical findings are, our recommendations. And the exciting thing is we can really predict, using quadratic regression, we can predict how long it's going to be before that person hits Alzheimer's disease. And since this is anatomical, they don't have to have the disease. This could be 5, maybe 10 years before they get it. Okay? So what we want to do is do the future of healthcare. An ounce of prevention is worth a pound of cure. And our device, Arethusa, bypasses that blockage. Okay? This is an early iteration. It looks a little different now, but it works the same way. As I said, it goes up through the nose and gets implanted into the cribriform plate. This is what it looks like here in an actual human skull. From the brain side, you can see it poking through. Oops. Our neurosurgeons have put this into 4 cadavers, and they say that they can put it into a human in about 5 minutes, into a patient, that is, in about 5 minutes under twilight anesthesia, which is what you would be under for like a wisdom tooth extraction. So it's very amenable and it's also accessible. So if something bad were to happen, some adverse effect, it could easily be taken out. So that's our plan. The Procogni app, we're improving it all the time. We're adding AI-driven questions that can help guide users toward better health, better memory health. It's globally available. It could be used in for healthcare provider networks that have thousands or millions of subscribers, and the response is immediate. So they can really track a subject over time. They can do it in the office. The person can do it on their iPhone every day or week. I'm actually at the Alzheimer's conference in Boston right now, and there are some people talking about how great it would be to assess Alzheimer's subjects every month or every week. And we can do that with the Procogni app. Okay, the diagnostic aspect of this is also a good way for healthcare systems to get a handle on their aging populations. With the silver tsunami, some places, for example, Japan, 25% of the population is over 65, and it would be very helpful for them to know if their memory problems are going to progress to Alzheimer's or not. And this can be done for essentially the cost of a blood panel. So it's very manageable, and it can be done all across Central America and South America as well. Okay, lastly is our device, Arethusa, the brain drain, as Margaretta calls it. It's the, we want to do a clinical trial. We're going to do a clinical trial in the next 2 years with 20 subjects, and it should take 2 years to hit our primary endpoint, which is a reduction of mild cognitive impairment and stopping MCI, and compare that to subjects that didn't get it, and their MCI should continue to get worse and worse. And our second endpoint will be a few years later, where the prevalence, I should say, of Alzheimer's disease in the untreated subjects should be much higher than in our treated subjects, and none of them should really get Alzheimer's disease if we're correct about this. So that's it. I don't work alone. I'd just like to thank our team, and including our newest advisor, Margaretta. All right. Thank you, Margaretta.
Thank you so much, Doug. That was a fantastic presentation. And Jean and Greg and Doug, unfortunately, we're at the end of our hour, and we need to go on to our next panel, which is all about investing in longevity. So we're not going to have time for our discussion today, but our next summit, we're going to leave more time, so we'll be able to have a discussion. And I'm inviting you all back for our next summit. So thank you all very, very much.
Thank you, Margaretta. Thank you, everybody.